Well, thanks for inviting. Good morning. I apologize for my English because I talk in Portuguese, my native language. Well, our talk is about Sindecan. One moment, please. One moment. It's okay. It's okay. Moment. Yes. Our talk is about Syndicam 4 as a regulator for anoic resistance. Anoics is a type cell death that occurs due to the cell's loss of adherence to its matrix. It's an important physiological process as it prevents cells that die from their matrix to survive and colonize different tissues. Anoic-resistant cells are able to survive and after loss of adhesion and thus colonize other tissues. This process is called metastasis. There are several causes that lead to the acquisition of anoic resistance, such as genetic instability, intertumoral hypoxia, EMT activation, expression of stimulus makers, and the others. These cells modify their cell signaling, promote growth, survival, and metabolic dysregulation. Thus, cells can evade the primary tumor, travel through the vascular system, and colonize the secondary sites, giving rise to new tumors. We have been working with anoic-resistant endothelial cells. Heavy-arched endothelial cells were to be cultured in a plate treated with 1% agarose for 96 hours. The agarose coat prevents the cell adhesion. In this way, most of the cells died. However, some cells managed to bypass the anoic process and survive. Then, these cells were cultured under ideal conditions and were subjected to adhesion blocking again. These adhesion blocking cycles were repeated 5 times. So we get anoic resistant endothelial cells. This experiment model was characterized in these 2 papers from our group. The first, acquisition of anoic-resistant upregulation syndicam-4 expression and endothelial cells. Second, acquisition of anoic-resistance promotes alterations in the RAS and PI3K signal pathways and matrix hemodilian in endothelial cells. This first publication showed that anoic-resistant cells showed high proliferation rate, low adhesion to fibronectin, laminin, and collagen 4 substrates, high invasive capacity, and low apoptosis rate. One moment, please. Yes, and the low apoptosis rate is similar parental cells, EC parental cells. Observing these changes, we sought to understand what change occurred in cell signals pathways that favor the acquisition of these characteristics. We also observed one increase in cells in the S phase, here, here, and here, similar to EJ-Has oncogenic cells. Observing this change, we sought to understand what change occurred in cell signal pathways that favored the acquisition of this characteristic. Thus, we decided to investigate the expression of the heparan sulfate proteoglycan syndecan 4 in anoix-resistant endothelial cells. We have seen that there is one increase in the syndecan 4 expression in anoix-resistant endothelial cells. We also observed one increase in the synthesis of sulfatate glycosaminoglycans, heparan sulfate, and chondroitin sulfate, synthesized by anoix-resistant cells. In cell extract, one increase, and medium culture, two increase. In the other paper, acquisition of anoix resistance promotes alteration in the Has and PI3K signaling pathways and matrix remodeling in endothelial cells. Then, now we investigated the expression of proteins involved in signaling in the Has and PI3K pathways. These pathways are associated with cell survival, growth, proliferation, and cell motility. In the other publication, the results show high expression of Has, high expression of ERK, high expression of PI3K, and AKT. Both high expression in endothelial cells are not resistant to anoix. In summary, anoix-resistant endothelial cells present one increase in the expression of integrins, one decrease in the expression of fibronectin, and an increase of the expression metalloproteases, perlecan proteoglycan, and heparanase enzyme. In particular, we observed one increase in the express of syndecan 4 proteoglycan, the target of the study. Syndecan 4 acts as a co-receptor for extracellular matrix molecules and growth factors. Thus, Syndicant-4 participates in several cell signaling pathways that trigger cellular events such as proliferation, adhesion, cell survival. Several works have studied the expression of syndicant-4 in different types of cancer, such as breast cancer overexpressed, colorectal reduced, glioma overexpressed, liver overexpressed, melanoma overexpressed, neuroblastoma reduced, osteosarcoma overexpressed, testicular overexpressed, popular thyroid carcinoma overexpression, kidney overexpressed, and bladder overexpression. And also the effects the anti-cancer drugs and syndicant-4 expression in different cells models. And trastuzumab, the cell type endothelial cells, is affected decreases syndicant-4 expression. Panitumumab and cell type colon cancer decreases syndicant-4 expression. And ESCO, breast cancer and syndicant-4 regulation, upregulation. This data is published in our paper, syndicant-4 as pathogenesis factor and therapeutic target in cancer. So we decided to investigate the role of the syndicant-4 in anoix resistance. Thus, we silence the syndicant-4 gene in anoix-resistant endothelial cells. This recent publication, syndicant-4 RNA, inhibits syndicant-4 gene and protein expression in anoix-resistant cells. After syndicant-4 gene silence, the cells showed decrease in syndicant-4 gene and protein expression to compare the parental cells anoix resistance. The silent cells also showed a decrease in the expression sulfatases glycosaminoglycans, heparan sulfate, and chondroitin sulfate, and cellular structure, and culture medium. The silence of syndicant-4 also altered the proliferation and apoptosis rates in anoix-resistant cells. We observed that the silent cells showed a growth rate significantly lower than the parental cell lines. This parental cell line is ADA1EC. The similar wild type. In relation to apoptosis, the results obtained showed a significant increase in apoptosis rate in silent cells, similar to wild cells. The cells present a behavior similar to the wild endothelial cells. The silent cells present in behavior similar to the wild endothelial cells. Besides, here we showed that after syndecant-4 silence, the anoix-resistant endothelial cells showed significantly higher collagen and laminin adhesive capacity, this here, and lower invasive capacity compared to the parent endothelial cells. And again, silent cells present behavior similar to wide endothelial cells. And to investigate the role of syndecant-4 in angiogenesis, we analyzed the formation of tubes in gel by endothelial cells. Our results revealed one reduction of the angiogenesis in silent cells in relation to parental cells anoix resistant. Again, silent cells present behavior similar to the wide endothelial cells. In conclusion, the results present provides evidence to support the involvement of syndecant-4 in maintaining the transformative phenotype of anoix-resistant endothelial cells. We believe that the control modulation of the syndecant-4 expression could be applied as a therapeutic approach. Further studies are needed to understand the specific mechanisms that lead the syndecant-4 to reverse the transformative phenotype of anoix-resistant endothelial cells. Because the results obtained so far, we intended to study expression of molecules involved in the RAS, ERK, and PI3K, AKT signaling pathways, expression of cytoskeletal molecules, remodeling of this extracellular matrix, and expression of proteins involved in syndecant-4 biosynthesis. After, of course, syndecant-4 silence. Thanks for the attention.